All right, we're going to talk today about this thing that we call the central dogma. Okay, so our objective is to be able to tell me what the central dogma is, number one. The second thing is you need to be able to perform the different parts of the central dogma. Now, you have in your book a page that has the central dogma on it. It should be, in your book, it is number 50, right? Okay, page 50. So turn to that page. You should have that page filled out. The central dogma is that DNA undergoes transcription to RNA. The RNA, okay, is then translated into a protein. So, DNA to RNA to protein is the central dogma. It's very, very important in molecular biology. Now, let's talk a little bit about this process and where it's occurring at. This isn't part of your notes, but you're going to see it again, so maybe you want to jot it down where you got a little extra space. Okay? Here's a bacteria cell. Now, what do we know about bacteria cells? Okay? They do not have what? A nucleus, right? So, a bacteria cell is a prokaryotic cell. It does not have a nucleus. So, the DNA is transcribed into RNA, and then translation of the RNA into a protein. See where that says polypeptide and all those little purple spheres are mixed together? That is the protein that is being built. Where is the protein built at? The ribosome. Okay? If you remember way back when we talked about the cell, we said that the ribosomes were where protein synthesis occurs. It's a protein factory. So the ribosomes are where we make the proteins. This polypeptide is just another word for a protein. Many peptides. Now, what we're going to see, folks, in eukaryotic cells, it's a little different. Why? Because eukaryotes have a nucleus. Are there any ribosomes inside the nucleus? Okay. So, what has to happen is we have DNA transcribed into messenger RNA. Since the DNA is in the nucleus, that's where that's got to happen at, right? Well, after we have the RNA, which in this case is mRNA, okay? Because the mRNA, remember we said it's like the blueprint, right? We said the other day it's like the blueprint, okay? So the mRNA then goes out into the cytoplasm, which is where we have the translation occurring, where the mRNA, see how this mRNA there goes into the ribosome and is read out, and then we have the polypeptide. Okay? So where is protein synthesis? In the cytoplasm. In the cytoplasm. Where is transcription occurring? In the nucleus. Okay? Now, here we go. This is what you have on for your notes over the central dogma. And remember, okay, DNA is transcribed into RNA, which is then translated into amino acids, into proteins. Okay? Now, if you remember when we talked about amino acids, okay, amino acids are the building blocks of proteins. You remember how many amino acids there were? It's more than 4. Anybody? Anybody? Is amino acids in this book, or was that first semester? I think it's in first semester, okay? It's in first semester's book, which I have. But there are 20 amino acids. All right, so if you would count on that codon chart on 53, those are the amino acids. Okay, those are the amino acids. The very first one, which is UUU, phenylalanine. That would be one amino acid. So when we have the different building blocks of proteins are those 20 amino acids. Now, as I look up here, could you write the mRNA strand for that strand of DNA? Do you think you could write that? Go ahead and try it. Go ahead and try, write the RNA strand for that particular strand of DNA. And once you get that done, I will do mine up on the board here. Now, making this RNA strand is just simply working with Chargaff, right? What was Chargaff's rule? Do you remember? What did Chargaff say? If you don't remember, go back and look. Okay? Chargaff said what? Anyone? Okay? Lewis? A always pairs with T, C with G. Well, in this case, we don't have T's, do we? Because T's are replaced in RNA with a U. So an A will pair with a U. So I can see I have G, U, G, C, A, U, C, U, and G, right? Is that what you got? So, Jonathan, you could do it? Huh? So you could get it? All right. So, folks, what you have just done there is transcription. You've made that blueprint. All right? Now, in translation, what is happening is you need to read to determine what amino acid is coming in. Okay? Now, if you look on your codon chart, see where it says on the left-hand side, first base. In the middle, second base. And on the bottom, third base. Or excuse me, on the right, third base. Now, we're not talking first base and third base like a baseball field. We're talking about nitrogenous bases. Okay? So, right here is the chart that you have, right? Now, I want you to find my first base, which is, this is what we call a codon. A codon is a group of 3. So my codon is a group of 3. I look, what is G, U, G? I find it, I read G, I read U, I read G, and it is G right here, U right here, And finally, G right here. GUG is valine, right? So I just simply put valine. Now that will be bonded to my next codon. Okay? Sometimes these codons are referred to, folks, as triplets. Why do you think? Because there's 3 of them, right? There's 3 of them. So sometimes we hear these called triplets. Okay? CAU. Okay, so I'm going to erase my last one, and I'm going to find C for my first one, A for my second one, and U for my third one, right? So CAU is histidine, right? We good to go on that one? Now let's look at my third one. Let's look at my third one here. CUG is my third codon. CUG. I'll get these out of here. C U G. What is it? Leucine? Whoa. Right? Leucine? So, basically, folks, okay, basically, we have these codons, right? We have these codons, and I'm not a very good person that draws, okay? I'm not a very good person that draws, but I'm not a very good drawer. But what we have here, when I draw this out, oh, that's kind of funky. Okay? We have a codon right here, right? We said that this is a codon. Those 3, right? Now, what we have is we have transfer RNA. And let's say I've got my transfer RNA, and it reads for 3 different things. And I can tell you this is going to look more like a hydra than it does transfer RNA. But hey, we'll have that. But this transfer RNA, okay, is what will be coming in, and it'll come in, and it'll read the 3 codons, and it will bring with it a little amino acid, and I'm going to mark this one as a square. And that one would be valine. So it would read the codon there, the the transfer RNA. So this is tRNA, that little blue thing there. Now, here I've got another one that's reading okay, I've got another transfer RNA that's reading this one, and it's bringing along, we'll make this a triangle with the H, because it's bringing along histidine. Okay, histidine. Then I have a third one. a third tRNA that reads that, and let's say that this is a circle, and it brings along this leucine, right? So each one of those now, okay, we're going to have the square and a peptide bond bringing me with the triangle, and then we're going to have a peptide bond. So here, can you see how I'm building the protein? Okay, you see how I'm building the protein, and we're adding one protein or one amino acid at a time. Okay? So that is building my specific protein, one amino acid at a time. So once again, folks, this is the central dogma. The central dogma, remember, DNA is transcribed into RNA. RNA is then translated into a bunch of those, a bunch of them. What do we call? Polypeptide, or it says amino acid on your paper, right above that, a protein. Okay? Where it says amino acid on your paper, right above that, a protein. Okay? So right here, I want you to write, I'll write mine below it. Because what are amino acids? They are the building blocks of proteins. So if you were going to tell me what's the central dogma, could you do it? Because believe me, folks, you're going to have to. Could you tell me the central dogma? Hopefully, I get a thumbs up there. Okay? Could you take a strand of DNA and make RNA from it? Could you then translate that into the amino acids if I gave you a chart? Okay? Not real tough, is it? Reading a chart? Hopefully not. But that's all the central dogma is. That's it. That's it.